Hello everyone, my name is Sara Gonzalez and I'm gonna talk about this article called Investigation of an Outbreak of Burkholderia cepacia infection caused by drug contamination in a tertiary hospital in China. Burkholderia cepacia is an aerobic non-fermenting gram-negative bacillus. As we all know, gram-negative bacillus are characterized by its cell envelope, which is conformed by two membrane cells and one thin wall of peptidoglycans. This bacilli is rich in wet soil, in water, in plants, in tropical environments, and also it has been found in hospitals. Many studies have reported outbreaks of nosocomial B. cepacia infections caused by the contamination of drugs or devices. The general objective of this article is to investigate an outbreak of Burkholderia cepacia infection caused by drug contamination in a tertiary hospital in China.
Methods. The first method used was the microbiology investigation. When the first case was reported, the drug was believed to be the source of infection. The hospital infection control and prevention staff used sterile swabs with sterile saline solution to collect samples from the environment, object surfaces, and hands. Moreover, samples of unopened drugs were sent for microbiologic testing. All the bacteria were isolated by using the following methods.
The second method is the PFGE. The pooled-field gel electrophoresis is a laboratory technique used by scientists to produce DNA fingerprint for a bacterial isolate. So the process consists in 5 steps. The first one is the bacterial culture. So the scientists take bacterial cells from an agar plate. Then the scientists mix the bacterial cells with melted agarose and pulls it into a plug mod. After that, the bacterial cells are broken open with biochemicals or lysed so that the DNA is free in the agarose plug. Then the scientist loads the DNA gelatin plug into a gel and places it into the electric field that separates the DNA fragments according to their size. And finally, the gel is stained so that the DNA can be seen under ultraviolet light. A digital camera takes the photograph of the gel and stores the picture in a computer. The DNA fragments produce a DNA fingerprint with a specific pattern. So the figure shows an example for an agarose gel where each lane represents a DNA fingerprint or pattern. Once a DNA fingerprint is created, they investigate the pattern to see if it is causing an outbreak or it is part of an ongoing outbreak.
The third method is VTEC. It is a system that uses cards with colorimetric reagents which are inoculated with the suspension of a pure microbial culture, and the development profile is interpreted automatically by an optical density system. Reactive cards have 62 wells that each contain an individual test substrate. With this substrate there are several metabolic activities such as acidification, alkalinization, enzymatic hydrolysis, and development in the presence of inhibitory substances. These cards have barcodes that contain information on the type of product, lot number, expiration date, and a unique identifier that can be linked to the sample either before or after the card is loaded into the systems. There are 4 types of reactive cards available for the identification of different organisms. The first one is the GN. to identify the gram-negative bacilli fermenters and non-fermenters. The second one is the GP for gram-positive Kochian bacilli forming bacilli. The third is the YST for yeast and yeast organisms, and the last one is the BCL for gram-positive spore forming bacilli. It is used for microbial identification and antibiotic sensibility tests.
The fourth and last method was the susceptibility test. With this test, the discs are impregnated with various defined concentrations of different antibiotics and they are placed into the agar gel. After incubation that can take from 16 to 24 hours at 35 centigrades, zones of growth inhibition around each of the antibiotic discs are measured in their nearest millimeters. A clear circular zone of no growth in the immediate vicinity of a disc indicates susceptibility to that antimicrobial. The size of zone can be related to the MIC and results recorded as whether the organism is susceptible, intermediate susceptible, or resistant to that antibiotic. So this test was used in this article to know if Burkholdeриa's isolates were susceptible or resistant.
Now the results. In the table 1, it shows that among surgical instruments, B. cepacia was isolated from 2 of the 10 grills used for perineal prostate biopsy. The positive rate was 20%, and 8 samples of an open analgesic gel were obtained from 5 different batches, of which 2 were positive for B. cepacia. The positive rate was 100%.
This epidemiologic curve relates the total number of cases of infection and the date of culture. A total of 9 patients in the urology ward were positive for B. cepacia from January 2017 to March 2018. The incidence rate of nosocomial infection from January 2017 to October 2017 was the 0%. But from November 2017 to March 2018, the incidence rate was a 0.43%, thereby confirming the outbreak.
Table number 3 shows that median patient age was 65 years old. All patients had undergone a surgery before symptom onset, and B. cepacia was isolated from urine specimens of 9 patients, of which 2 showed B. cepacia in the blood.
The homology of bacteria isolated from patient specimens and analgesic gel was determined using pulse field gel electrophoresis. So here we can see that the M line was labeled. The line number 1 represents the specimens of patients without Burkholderia cepacia infections. The lines number 2 from number 11 represents the specimens of patients with Burkholderia cepacia infections, and the lines number 12 and 13 represents the analgesic gel samples. With this, we can confirm that all isolation patients and drug were homologous.
Discussion. The State Pharmacopoeia Committee said that there was no cepacia, but the article proved the opposite. Also, Peterson et al. Said that immunocompromised patients are primarily infected, and the article agrees. Finally, Morin et al. States that the presence of bacteria resistant to preservatives is due to product design defects that lead to improper disinfection, and the article agrees.
The conclusions are: the first one, the investigation revealed that the outbreak of Burkholderia cepacia infection was caused by drug contamination. And the second one is that Burkholderia cepacia should be added to the list of control bacteria for drug microbiologic monitoring under supervision.
Thank you so much for watching.